Hello, my name is Megan Hansen, and today I will be presenting on biological productivity in Foraminifera Microfossils, a fingerprint of past ocean and glacier conditions offshore Antarctica. This research is being done under the supervision of Dr. Rebecca Totten in the Department of Geological Sciences. I'd first like to start this presentation by discussing the background of Antarctic paleoclimate research and the necessity for projects in this area. We'll then discuss the Thor project, which is the initiative that's spearheading these projects. We'll then talk about my specific work in foraminifera assemblages in Cranton Bay, including methods and initial findings. So to first put this research into context, Antarctic paleoclimate history is becoming more and more relevant as you move through the 21st century due to the warming of our global oceans at an alarming rate. I'm sure most of you have seen the global atmospheric temperature graph that shows the unprecedented temperature spikes that we're seeing since the Industrial Revolution. Our oceans are seeing a very similar pattern, leading to the melting of many of our massive ice shelves in Antarctica. This is a particularly evident pattern off the coast of West Antarctica, which you can see in the figure to the left. The purple line represents the massive ice shelf loss that we've experienced from 1980 to 2018, and the plummets evident at negative 64.8 gigatons per year, which is very significant for just a 40-year time period. On the right, you can see a similar trend illustrated throughout the entirety of the Antarctic ice sheet. We're losing about 125 gigatons per year on average throughout all of Antarctica. And a particular focus of recent Antarctica research is on the Thwaites Glacier, otherwise known as the Doomsday Glacier, which is a large controller on the stability of the entire West Antarctic ice shelf. The Amundsen Sea is an embayment that borders both Thwaites Glacier and the Pine Islands Glacier, and its oceanographic conditions, particularly the presence of circumpolar deep water, or CDW, largely influence the melting patterns of these two glaciers. Now, CDW is a relatively warm water circulation that leads to basal melting and overall instability of the ice shelf, and this is indicated by the red path seen in the figure. Ultimately, our goal with this research is to construct an extensive multi-proxy analysis of sediment cores that will help us get an idea of how the West Antarctic ice shelf has responded to past oceanographic conditions. With this knowledge, we'll be able to get an idea of how our ice shelves may continue to respond to our rapidly changing oceanic and atmospheric conditions in modern times. The driving force behind this research is the International Thwaites Glacier Collaboration, or ITGC, which is formed as a 5-year initiative targeting research in the Amundsen Sea offshore Thwaites Glacier. The project was jointly funded by the U.S. National Science Foundation and the U.K. Natural Environment Research Council, and it includes collaborators from both the United States and the United Kingdom. The Thor project under this group is specifically investigating the paleoclimatic history of Thwaites Glacier pre-satellite imagery, with hopes to use historical reconstruction of the Holocene impact climate. Sediment cores were collected offshore Thwaites Glacier and throughout the Amundsen Sea in 2019 and again in 2020 and 2022 on a research expedition involving the ITGC team. And these have been used in proxy analyses by the various institutions involved in the project since then. Now here we have a map detailing the area of West Antarctica where I will be focusing my research on. This is the eastern area of the Amundsen Sea embayment, and the research expedition in 2019 visited multiple different sites off different ice shelves, but the 3 cores that I'll be focusing on are all within Cranton Bay, and you can see them located in the top left with the scope of all of Antarctica and more up close in the bottom right. Now, Cranton Bay is a relatively small and shallow embayment within the Amundsen Sea, and deglaciated at least at the early Holocene, which is thought to be an earlier deglaciation than the rest of the Amundsen Sea. The history of sediment deposition and primary productivity since this point have all been preserved in the bay and can indicate the oceanographic conditions throughout this time period. The area was left virtually unexplored up until the 2019 research expedition by ITGC, and recent research in this area has shown a shocking peak in primary productivity with the possibility of high concentration of zooplankton microfossils, or foraminifera, which can provide an in-depth analysis of the melting patterns that may have occurred. Now, the core that I'll be focusing on mainly is KC27, which is the last casting core that was taken on the 2019 expedition. So here you can see a line scan and a CT scan taken of the KC27 core. And as shown earlier, this core is close to the coast of Cranton Bay, and it's neighbored by JGC74 and MC73 cores, which will be analyzed in conjunction to further the depth of our data. After a precursory look at general foraminiferal patterns using the leftover sample in large increments from the 2019 research expedition, we further sampled the KC27 core in higher resolution in September at the Marine Geology Repository at Oregon State University. The samples were taken in 2-centimeter increments every 10 centimeters throughout the core, and then the samples were transported back to UA for further analysis. CT scans were also used to locate regions of interest, which you can see zoomed in at the top. So bioturbation, or disturbance of the sediments by living organisms, is seen throughout much of the top 170 centimeters of the core. Laminations were present in the second half of the core with less bioturbation, indicating that these sediments are probably more preserved in the place where they were deposited. The ultimate goal is to conduct a multi-proxy analysis on this core using foraminifera and diatom assemblages, grain size analysis, radiocarbon dating, and carbon and nitrogen stable isotopes. My primary goal at this point in the project, however, is focused on foraminifera assemblages. Now, what are foraminifera? Foraminifera, or forams, are microscopic single-celled organisms with external tester shells, typically made of calcium carbonate, which form microfossils that preserve well on the seafloor in the sediments. These can be used for radiocarbon dating or for foraminifera assemblages that we're currently focusing on. On the right, you can see some of the forams that I've been picking from the KC27 core. The bottom picture is a collection of Globocastagelina foraminifera, which is one of the most prevalent species we've found to date in this core. The top picture is also of a planktic foraminifera called Neoglobopachyderma. Now, there's a couple different types of foraminifera that are found in Antarctic sediments, as I mentioned. The two of these main groups are benthic and planktic foraminifera. So benthic foraminifera are typically used for radiocarbon dating. As they live on the seafloor, so they're preserved more accurately where they died, whereas planktic foraminifera kind of live in the water column, so their locations in the sediment record are a little bit more variable. However, both types are incredibly relevant for foraminifera assemblages, and for these species are differentiated and counted into plates, and from this we can determine their prevalence at a certain time in the sediment record. Since collecting our most recent samples, I've sieved through the core every 10 centimeters using hexametaphosphate to deflocculate clay particles, and sieving is done with a 63 micrometer sieve to isolate relatively larger particles without the loss of foraminifera, which are generally larger than this. Sieving each sample makes it possible to locate foraminifera without the clay particles kind of sticking all the sediment together and making it challenging to pick apart. After the samples are fully dried, they can be split into halves or fourths depending on the volume of the sample, and this increases the efficiency of the picking process. As for the picking process, we use microscopes and look through the sediments to try to find forams and then separate them by species and count them into microscope slides. And then at this point, previously published literature typically requires around 300 foraminifera to be picked in a sample for multivariate statistical analysis to be valid, and plates can also be sent for radiocarbon analysis at this point if desired, and we currently have 2 samples sent to UC Irvine for analysis. Preliminary analysis of the core has shown an unexpectedly high amount of primary productivity, including foraminifera, diatoms, sponge spicules, and dark green organic matter. At this point in the project, we're not far enough along to conduct a complete assemblage. However, initial picking has allowed us to determine prevalent species at 2 of the intervals of the core. So here we have 4 of the species of foraminifera. I found the 2 intervals that I've picked, 265 to 267 centimeters and 155 to 157 centimeters. As I mentioned previously, Globocastagelina, which you can see second to the left, is one of the most common species we've found. And this is a benthic species of foraminifera that's generally indicative of the presence of CDW leading to the basal melting we were talking about earlier. We've also found a notable presence of these 3 other species, which are indicative of just general high primary productivity. Arenaceous foraminifera have also been found in high numbers, mainly in the 155 to 157 centimeter sample, but notably present in both. Planktic foraminifera, particularly Neoglobopachyderma, seen on the left, have been found in high numbers at the higher interval, but not lower in the core. While further data collection is necessary, high productivity, it looks promising for both the low parts and the middle of the core for productive conditions during the Holocene. As this project progresses, we'll be able to get a better idea and track these conditions at this point and through time until present day. I'd like to acknowledge our funding agencies and the International Thwaites Glacier collaboration along with the Totten Lab for their support in this project. These are my references, and are there any questions?
Hi, Megan. Are there any uncertainties or limitations of foraminifera as a proxy that you should take into account when using them for a paleoclimate reconstruction?
Yeah, absolutely. So there's, of course, always bumps that you can run into in research. Currently, an issue we're having is you need, as I mentioned, a lot of foraminifera in samples to be able to make conclusions about their locations. So we're having an issue with this with the preservation of the core. Sometimes you don't find as many foraminifera as you need. You also have issues, as I mentioned, with using planktic foraminifera as they can drift around in the sediment column. Also with bioturbation, it's kind of a harder it's harder to determine exactly where the sediments were deposited. So to kind of remedy this, we use multiple different proxies, as I was mentioning. So the grain size analysis, the isotope analysis, and this kind of lets us corroborate all our results at once and kind of feel more confident about what we're deciding on.
How many additional cores were taken on the research expedition, and are all of them undergoing a similar analysis and comparison process?
Yeah, so the core that I'm focusing on is from the 2019 research expedition, which had 28 total cores, and these are all kept at Oregon State, and all of the institutions involved in the project all across the US and the UK are using these in different projects. They're not all the same as my project, but they're all being used for paleoclimate reconstruction pretty much.
How do you decide on the resolution and location of the samples you are taking from each core?
Yeah, so for this project, we wanted to get a higher resolution of samples to take from the core to just have a more solid idea of where things were. With the bigger intervals, it was a little bit more challenging to isolate what was going on. However, we do have to be careful with the sediment cores, as they're shared across institutions. You can't take too much sample from them, so we had to be careful to not take all of the sample from the core, but still get everything that we needed for the project. So we decided on 10 centimeter increments to kind of just standardize the process, but then it's not oversampling the core.